So today's lecture will be on the muscular system. Specifically, we'll be focusing mostly on skeletal muscle because the cardiac system will have its own lecture, which will focus more on cardiac muscle, and smooth muscle will be in the other lectures as well, and like the other systems like the GI tract and the renal system. Okay. But I will be going over the other types of muscle as well today, briefly in the beginning. As a personal introduction, if this is your first lecture with me, my name is Mark Abdurrahmanov. I'm a graduate of Stony Brook University with a Bachelor of Science in Biochemistry. My MCAT date was this January, and I scored 520. This is my email if you ever want to contact me with any questions regarding the course, the MCAT, or any topics. Quick description of the course. We have 3 lectures per week, Thursday, Friday, and Saturday. This is the last one for this week. Every Sunday at 8:30 p.m. Eastern Standard Time, we'll also be holding office hours. We'll be covering, throughout this course, we'll be covering most of the highest yield MCAT content, and all lectures will begin at 8:30 p.m. Eastern Standard Time. Okay. This is a link to GroupMe, and actually, Janelle, the other tutor, will post it in the chat as well. The GroupMe is a place where we post all the lecture recording links and PowerPoints after each lecture. We also post any updates or announcements to scheduling there. It's also a great way to just ask us questions, me or any of the other tutors. And we have a lot of students there who may be able to help you with anything as well, right? Either finding resources or answering questions you may have. So what are the learning objectives for today? So first, what are the different types of muscle? I already covered it a little bit in the beginning, but we'll be going in depth into the different types. What makes up a sarcomere and what is a sarcomere? We'll also be learning how do muscles begin contraction, what is the myosin-actin cycle, and we'll also be talking about how muscles terminate contraction. Okay. Okay. So let's move on to the content. Firstly, we're going to be just do a general overview on muscles as a whole. So what are the different muscle types? So there are 3 types in our bodies. There's skeletal, cardiac, and smooth. Each type is used in different systems and for different purposes. Like I mentioned, today we'll be mainly focusing on skeletal, as the other 2 will come up in other lectures. But I want to show you what the difference is between all 3. So all the way on the left, you have smooth muscle. The reason it's called smooth muscle is because it's the only muscle type of the 3 that's not striated. You can see right here, not striated. So when you look at it, it actually looks smooth. Here's like a picture of it under a microscope. Whereas if you look at these 2 types of muscles, they have these little like lines over it. Those are called striations, and that's why they're referred to as striated, whereas smooth muscle is not. Smooth muscle mainly is found in walls of internal organs, as you can see here, in your GI tract, in your kidneys, in your liver, everywhere, most of your organs, the walls, and even in your blood vessels, the walls have smooth muscle. Okay. The muscles are controlled involuntarily, just like you don't voluntarily control whether or not your stomach expands or contracts, or whether your blood vessels contract or expand voluntarily. Your body does that automatically for you, right? Next is cardiac muscle, and as the name suggests, this is muscle that only is found in the heart, right? It's unlike in smooth muscle, it's striated as well, right? So it has these little lines on it. And just like smooth muscle, it's involuntary, meaning that you don't control how fast your heart beats or contracts. Your body does that automatically for you. Lastly, skeletal muscle, which is what we're going to be talking about mostly today. It's striated. The fibers are striated, meaning, again, they have these lines, these striations. It's usually attached to your skeleton. So, for example, if you can see here, this looks like a leg, right? The muscles are actually attached to the bones of the leg, right? And they pull on the bones. That's why it's called skeletal muscle. And the 2 unique things about skeletal muscle is, first, unlike the other types, it's voluntary. You choose when you want to like move your arm or move your leg or move your head or anything like that. You choose how you can voluntarily move these muscles at any time. And the other difference is that the muscle fibers in skeletal muscle are multinucleated, meaning if you can see here, there's these muscle fibers, they have multiple nucleuses per fiber, whereas the other two types, if you can see here, are uninucleated, meaning they only have one nucleus per fiber, okay? I would recommend actually memorizing this like picture or the information that's present in this picture, mainly each type of muscle, whether it's striated or not striated, whether it's voluntary or involuntary, where each muscle is found, so smooth muscle and organs, cardiac muscle in the heart, and skeletal muscle attached to skeleton. Those are the main things I would focus on, right? In terms of differentiating between the different types of muscle. A lot of different things for muscle contraction are actually fairly similar for all types of muscle. So you would be able to like transfer some knowledge between, okay? But let's move on to skeletal muscle. So this is what the diagram for skeletal muscle looks like. So as you can see, this is a muscle fiber, right? It's this long tube and there are the striations here with the lines. Sorry about that. And as I mentioned, multinucleated. So you can see there's nucleuses everywhere here, right? And obviously mitochondria all around to provide energy. The sarcolemma is kind of like the membrane, right? The membrane for a muscle fiber. And there's these things called myofibrils, these little smaller tubes inside this big tube, right? And we can actually see a close-up of it right here. Here we can also see the sarcoplasmic reticulum, which is very similar to the endoplasmic reticulum in any other cell, except in muscle cells, it's called the sarcoplasmic reticulum. If you notice, everything in muscle cells is just called sarco something, sarcoplasmic reticulum, sarcolemma, right? That just signifies that it's part of the muscle. So you notice there's in a myofibril, and this is something that we'll be covering a lot, there's this thing called a sarcomere, right? If you notice there's this Z disc right here, this green line, and then a Z disc on this side, and there's many of these on a myofibril and many more in a muscle fiber. The length between one Z disc to another Z disc is called a sarcomere. It's a unit of measurement, and in making up a sarcomere are different types of filaments. So you can see here, these dark purple ones are called thick filaments or myosin, and these thin ones are called thin filaments or actin. And as well as there's these, we break up this big unit, the sarcomere, into smaller units called the I band. Sorry, my mouse. The I band, the A band, the H zone. Over here, there's this M line, and actually it's right here as well. But this is for a different sarcomere. So if you had another Z band right here, this would be the M line for that sarcomere. But for this sarcomere, the M line is this these three lines here. It's the midpoint, right? So as shown in the previous slide, a sarcomere is a unit of a myofibril from Z disc to Z disc. It's how a muscle contraction occurs. During muscle contraction, sarcomere length decreases. So if you want to think of this like sarcomere, when muscles contract, the myofibril contracts, and actually specifically the sarcomere itself gets shorter, right? Gets contracted. There are four components of a sarcomere that you should know: the I band, the A band, the H zone, and the M line, as well as the Z line, right? A quick way I memorized this is, you know that in order it goes I, A, H, M, and then from both sides, so it would be H, A, I, right? This is what like a sarcomere, but really you just have to know IHM, right? I am, and that's a good way to memorize like from left to right or from right to left, going from the outside to the inside, the direction, the order of the different bands or zones. Okay, I am. Okay. Now, again, I'm showing a picture of a sarcomere, right? These are the Z lines at the edges, the Z discs. The M line here in the middle is not shown, but this big red thing is the myosin, the thick filament, and these little blue lines are the thin filament, and these are the zones, okay? So in the I band, it's only thin filament. So if you see here, the I band ends right here. There's only these blue lines. There's none of these red thick filaments. In the A band, which is next, there's both these blue lines and these red thick filaments. In the H zone, there's only these red thick filaments. There's none of these blue lines, okay? An important note is that during contraction, only the thin filament moves and actually contracts. Thick filament doesn't. This means that while during contraction, sarcomere length, H zone length, and I band length decreases, A band length doesn't. So let's explain that. If you notice, an A band is from here to here. This is actually the whole length of the thick filament, right? So if you see this red line, the A band is always the length of the thick filament. If the thick filament never gets shorter because it doesn't contract, the A band doesn't get shorter during muscle contraction, right? Whereas the thin filament does. So if you want to think of it, the thin filament, the blue, is going this way and this way. Eventually, all of this H zone, if this moves closer to here and this moves closer to here, the H zone will only be this small part that hasn't been reached yet, this new part. The H zone will have gone from this size to this size, right? So it would have shrunk. Similarly, with the A band is only where the thin filament is. Well, if the thin filament and the walls, these black bars, the Z discs, move closer to here, well, now the A band is only this length rather than this length that it initially was, right? Right, so it moves close together, and the overlap, as a muscle contracts, the overlap between thin filaments and thick filaments gets bigger. And a way to easily memorize which filaments decrease in length is the ones that have only one type decrease, because the overlap gets bigger. Whereas something like the A band, which has overlap, it has both, doesn't change in size, okay? Any questions before I move on after this brief overview? Because I believe, oh, actually here, I wanted to show you this image beforehand. So this is a relaxed muscle and this is a contracted muscle on the bottom. The blue stuff is the thin filaments, with this red being the thick filaments, right? And everything else is labeled for you. If you notice, the I band here is from here to here, right? Whereas in the final product, it's only like this. So it's got shorter. The H zone initially was from here to here. And then in the end, it's only here to here, right? The whole sarcomere used to be like this. Now it's only like this, got smaller. Whereas the A band used to be from here to here. And now it's still from here to here, right? It's the same length. Just as a heads up, only the blue stuff moved closer in, right? Along with the walls. Okay, so somebody asked during athletics when they have cramps, what is going on? So that's a little bit, we'll cover that later at the end of the lecture, because that actually involves a lot more stuff, but it's basically, it's the same, kind of like the same thing when you have like twitches. It's just over contraction, right? So like if you look at this, the contraction, this is almost complete contraction, because you notice how there's almost no more room for the blue stuff, for the blue thin filaments to go. They're almost max contracted. Whenever you contract a muscle, that's not always the case. For example, just moving my arm like this, it probably is not causing this much contraction. It's causing very small amounts of contraction because you don't need much more. Whereas if you're lifting, like if I'm lifting like a 200-pound dumbbell and trying to curls with it or something, you would maximally contract, and you would have like to the point where it's actually like painful. Your muscles are pushing, like there's no more room in the middle here for the thin filaments to continue to contract. Okay. So I wanted to ask this question. I'm going to open up a poll. And I just wanted to see if you guys can answer this. Which of the following parts of the sarcomere does not change dimensions, which means just length or size during the course of muscle contraction? So please take some time to answer this. Okay, please take another minute or so to answer this question. And get your answers in. I'm going to end polling. Okay. Okay, so the results, the majority of people picked answer A, let's choice A, the H zone, with some also picking none of the above. So unfortunately, the correct answer is actually B, and nobody's picked it, and I think I know the reason why, right? So when you think of the Z line, you think of the length of the sarcomere, right? That's why you didn't pick Z, because I mentioned that the whole length of the sarcomere shifts. So here, right? You think this is the length of the Z line, and now it's here, and now it's smaller, right? Because these are the Z lines, right? But the Z line, this is not the Z line. This is the sarcomere length. The Z line is this. This is a Z line. This is a Z line. And then in the end, this is a Z line. This is a Z line. So this was a trick question, right? Because look, this length to this length, it didn't change, right? The actual wall itself didn't change length. The distance from this wall to the next wall changed length, but that's not what the answer choice was at. That's not what the answer choice was saying, right? It was just asking for the length of the Z line if it changed, and it does not. Most people pick choice A, which is the H zone. Remember, the H zone is the third zone. So the first zone is thin. The second zone is both. The third zone is the H zone. It's only thick filaments, right? Thick filaments, while thick filaments themselves don't move, this space gets filled up by the thin filaments. Look, in this zone where I drew this line, the thin filaments kind of pushed into it, right? And since the H zone is where only thick filaments are, all you're left with in an H zone in this final is this little tiny piece where there's no blue stuff, right? And you see how much smaller it's got from relaxed state to contracted state, right? Same thing with the I band. Initially start off with there's only this blue stuff, none of this red thick filament, right? And in the end, the only part that doesn't have this thick filament is like right here, right on the edges. So it got much smaller, right? Another component, as I mentioned, that will not change length, but it wasn't an answer choice, but it also would have been a correct answer if it was, is the A zone. The A zone is from here to here, right? And in here, it's from here to here, right? And you can see it's exactly the same length in both. Does that make sense, or do you want me to go over it? So this was a trick question, yeah. And the MCAT will try and trick you like this. Any questions? Okay, if there's no questions, I think we can move on. And feel free to type in the chat if you have any questions or raise your hand or anything. Okay. So we talked about the actual sarcomere. Now let's talk about the actual contraction. How does something contract? Right? How do muscles contract? And that's by this, the actin-myosin cross bridge. So as we mentioned during contraction, it is only actin which gets moved, not myosin. One way to think about the contraction is maybe think of myosin as a sort of motor, which works to contract the actin filament. So if you want to look at here, the myosin, these little red lines, they actually take these blue lines, the thin filaments, and they actually just push them together like a motor. They just keep pushing them closer and closer together, right? Like a motor would, right? Okay. The way that it does this is in a cycle where one myosin head binds to one actin, and then it pushes it forward towards the midpoint or the midline, and then it unbinds from that myosin, from that actin, and then binds the next actin in line, and then pushes that one, and then binds again, and then pushes that one, binds again, and pushes that one. Essentially, if you want to, like, this is an actin filament, that my hand is an actin filament, and this is a myosin head, takes it, pushes it, takes the next one, pushes it, takes the next one, pushes it, and right. So the difference between actin-myosin, there's many differences. For example, you don't really have to know like that many. They're made up of different components. You don't have to know what the components are, right? Myosin it has these like little bulb heads, whereas actin doesn't. Actin has a myosin binding spot. So the actin itself has a binding site on it where it allows myosin to bind to it, right? But myosin also binds to actin, right? Actin also has, and we'll talk about this later, a tropomyosin binding site. So if you look at the thin filament is these blue balls, right? The tropomyosin is this orange like line that's going through it. It's like a rope or like a vine. It has spots for that, but there's like many differences. Myosin has ATP and ADP binding sites, et cetera, et cetera. There's many differences between them, but yeah. So let's talk about, okay, so the myosin is this red, and it's the one with this like little bulb head, right? It's the myosin head. And the actin is these blue balls like that are arranged in like a rope, right? Yes, it is the myosin that moves the actin, not the other way around. The thick filament moves the thin filament. Okay. So this is like the whole cycle of how it gets moved. Because it's a cycle, you can essentially start at any point, and it makes sense. It should make sense, right? So here it starts at the top and goes like this. Personally, I remember this cycle. I actually like to start this cycle here at the bottom, right? To me, like that seems like a better starting point. And I do recommend watching maybe an animated video. So I don't have one here, but Janelle looked in the chat an animated video to watch after this as well to help it make sense. But I'll just try my best to explain it. So I prefer to start it here on the bottom. Again, it's a cycle. It doesn't matter where you start or where you end. So it starts off like this. So the myosin is not bound to anything right now. Well, it's bound to ATP, right? It has an ATP attached to it. As you may know, muscle contraction, it takes up energy. It takes a lot of energy to contract your muscles, so it requires ATP. This is how it's involved. ATP is bound to the myosin head, but it's not bound to the actin, which is right here, this blue line of balls, right? What happens is the ATP gets hydrolyzed. ATP hydrolysis is a way to get energy from ATP. ATP itself is not a source of energy. The only way to use ATP as energy is when you hydrolyze it or break it down. It gets broken down into two components, ADP and inorganic phosphate, PI, through hydrolysis. What that does is that creates energy for the myosin, and then the myosin then uses that energy to attach to the actin, right? It gets cocked, so it's like in this like inactive state. You give it some energy, it like perks up, and then it has enough energy to actually bind to the actin, as you can see right here, right? To bound to this actin. One myosin head binds to one actin, so one red bulb binds to one blue ball, right? And it still has the ADP and the inorganic phosphate attached to it. Next, what happens is the inorganic phosphate gets released. So this little purple thing comes off, right? What that does is that causes the myosin head from, it's in this position, right? Attached to an actin, it then moves like this, right? And which is called a power stroke. And because it moves like this and it's attached to the actin will also move like this, right? It'll move to the left or to the right, depending on which side of the sarcomere it is. It's moved towards the middle, right? In this picture, it's going to the left, right? So it moves to the left. The actin goes to the left as a result. Then ADP gets released as well. ADP comes off. This makes the myosin actin binding very weak. It's still bound and it's still like won't come off on its own, but it's weakly bound, right? The binding is weak because no ADP. Then what happens is ATP comes in, rebinds to myosin while it's still attached. ATP comes in, it binds, and then it releases and becomes inactive, right? And then the cycle starts again. ATP hydrolyzes, it perks up, and then it binds to the next actin. So you see the myosin itself, because it hasn't moved, and it still goes straight up when it gets like perked up or activated. Because it goes straight up and you moved the actin it was bound to last time to the left, it now will be bound to the actin that's next to it or to the right of it, and it'll push it as well. And then it'll get to the next one, push it as well, get to the next one, push it as well. And there's, on a thick filament, there's many myosins. Like there's a whole row of myosins here that are all simultaneously attaching, pushing, attaching to the next one, pushing, attaching to the next one, pushing, right? Okay. And I'm gonna, now that I explained it, right, okay, somebody asked, when somebody dies, what part of the cycle causes the deceased bodies to be stiff? This is called rigor mortis. We'll actually get to that in a little bit, in a couple of slides, so hold off on that. But now in these next couple of slides, I just want to write down what I explained in words. So assuming we start with myosin bound to ATP and uncharged and unattached to actin, the first step is that ATP will hydrolyze into ADP and inorganic phosphate, which will provide energy to the myosin. Using that energy, myosin head will become charged and attached to the actin. Once attached, the inorganic phosphate will detach from the myosin. This will cause it to form a power stroke, which is the actual moving, right? Moving the actin closer to the M line or contracting the sarcomere. Then once the power stroke is complete, ADP will also detach from the myosin, causing it to be very weakly bound to actin, but still bound. A new ATP will become bound to the myosin head, causing it to detach from the actin and return to its starting position. And then the cycle begins again with the next actin in line. Okay. I will cover that now. Actually, I was going to cover it after the question, but actually I'll cover it now because I think it might help you with the question. So notice what happens to your levels of ATP or energy once you die. I want to ask that in the chat to anybody who has. What happens to your levels of ATP inside your cells when you die? What do you think happens if you know that? Yeah, exactly. Your levels of ATP would go down, or they would gone because your body's not producing anymore because you're dead. And notice what is ATP needed for in the cycle, right? A new ATP becomes bound to the myosin. So when ATP binds to the myosin, it causes, so it is used for the power stroke as well and to energize, right? But what is it used for first before it can even energize, right? A new ATP is needed to bind to myosin, causing it to detach from the actin and return to its starting position, right? So if you didn't have ATP and you didn't have this step, before you can even do this step and energize it, it has to do this step. ATP has to bind to the myosin to detach from the actin. If you don't, what would happen is you would attach, move it, or like move it towards the center, and then it would just be stuck here. The myosin wouldn't be able to detach from the actin, right? And the sarcomere would forever be shortened, right? Because it would have already pushed it, right? It would have contracted it a bit, right? So if it can't detach, that means the sarcomere can never relax, right? Because you need to attach ATP in order to get the myosin off. If you can't get the myosin off, you can't go back to the actin can't go back to its original position. If it can't go back into its original position, the muscle can never relax, right? So you need ATP to bind to myosin in order to relax it as well, right? You need to let it unbind the myosin head. If there's no ATP, you would forever be stuck in this position, right? And that would cause like the rigidity. Yeah, it's what rigor mortis is this. You'd forever be stuck in this form. Yeah, ATP first ATP detaches myosin and then it energizes. If you can't detach myosin, then you won't energize it either. But so you wouldn't, after you die, you wouldn't continue to like stiffen. So like you wouldn't contract more, but you would always be stuck in like that level of contraction. Well, not forever, because eventually rigor mortis stops. I don't know actually the duration of it. That's something you could probably look up, but it's not needed. Like the main thing they want you to know is how it happens, right? And they may ask you about it, right? That's actually a common question about muscles that they ask. So they say when you have no energy, or when you have a disease that prevents you from doing this, or maybe when you die and you have no more energy, which step of the cycle will you be stuck at, right? And this is one example. So rigor mortis, you'll be stuck at this step. For example, if they say somebody has a disease that prevents hydrolysis of ATP, which step would you be stuck at? You'd be stuck at this step because you wouldn't be able to hydrolyze to move on to this step. If they say there's a problem with inorganic phosphate and you couldn't detach it, you'd be forever stuck at this step before the power stroke, right? That's a common question they may ask you regarding this cycle specifically. Okay. Yeah, and it's actually common that they will ask something like that, right? And actually, I'll ask something similar right now. This is a fairly straightforward, simple question, just testing on whether or not you understood the process, the cycle. So really think back, what does the hydrolysis of ATP bound to myosin directly cause? Okay, please take another 30 seconds or a minute to answer this. While you guys finish up 1 second, I will return in a minute. And on the poem. Okay, so the majority of you picked A, with C being very close behind, so it's fairly even. The correct answer is actually C. I'm glad that you guys at least narrowed down to both of these are the roles of ATP in the cycle. So both A and B are the roles of ATP to release actin from the myosin head from the actin filament and reversible binding of the myosin to the actin filament. However, the binding of ATP causes A, right? Just simply binding to myosin causes A. But the hydrolysis of ATP, which is the breakdown of ATP into ADP and inorganic phosphate, causes C, right? So first, ATP binds, and causes A, release of the myosin head from the actin filament. Then it hydrolyzes, and then it causes binding of the myosin to another active actin filament, right? To the next one in line, right? I remember that hydrolysis is of ATP is the breakdown of ATP for energy, right? What do you need energy for? You need energy for the myosin to actually bind to the actin, right? And the reason it's not D, which nobody picked, but just in case I wanted to go over, irreversible binding, because the binding can be reversed by just simply removing the ATP and adding by adding in another ATP. So you can reverse it, and if it was irreversible, then you would have a problem in your body, right? This would be a big issue. So that's why it's not irreversible binding, it's reversible binding. Okay, is that clear? A and C are both roles of ATP in the cycle, but A is a result of binding of ATP, whereas C is a result of hydrolysis of ATP. Okay, any questions before I move on? Okay. If there are no questions, I want to move on to the next section, which is initiation of contraction. So we know that how your muscles contract, we know what the muscles are made of and all of that. Now, how do we begin contraction? How does your body know to start contraction? How do your muscles know how to start contraction? Well, especially for skeletal muscle, but actually also for smooth muscle as well, it's just slightly different because it's involuntary. But for skeletal muscle, it comes from the nervous system. The main way that the nervous system communicates with the body is through action potentials and signaling molecules, which you may remember from our lecture on the nervous system, which we did a couple weeks ago. Initiation of the muscle contraction begins in the nervous system, and it contains both of these methods of communication, both action potentials and signaling molecules. Okay, so this diagram shows how it goes from the nervous system to a muscle cell. So this is an axon terminal, right? And it says right here, axon terminal. An axon terminal is the end of an axon. If you remember what an axon is, it's basically like a long, like a nerve tract or whatever. If you want to like in general terms, how you want to think of it, like in non-biology terms, it's kind of like the transport system from your brain to the rest of your body, right? And this is the end of it, the axon terminal, so like the tip. An action potential, you remember, it goes down an axon, so it would have traveled a long, long way, and then it would have finally, the action potential would have come in through here, right? Once the action potential reaches this end part, it goes to these voltage-gated calcium channels right here, right? These little pink dots, right? When the action potential reaches that point, the calcium channels open, and this portion gets flooded with calcium from this outside space. When this outside space gets flooded with calcium, this little bubble with these like green molecules inside goes from here to here to here, and then it finally reaches the end, and it actually opens and spills its contents out into this in-between space, which is called the synaptic cleft. Right? And this little green molecule is important. It's called acetylcholine, which I'll write down for you in a bit, but it's very important. It's called acetylcholine. And this is called a postsynaptic cleft. It's actually, this is on a muscle already. This is on the sarcolemma, which is like the membrane of a muscle fiber. And the sarcolemma has these little receptors, these little blue things, right? That react to acetylcholines. Whenever they sense acetylcholine, they activate. And what they do is they activate this little orange channel. This orange channel is also is a sodium channel. And what it does is when it's activated, it actually allows sodium to flow into the cell. Once sodium flows into the cell, it begins an action potential here that flows down through the muscle fiber. Okay. Okay. Somebody asked, the action potential uses sodium, but the other contraction uses Ca2. So we'll cover this in a second, but different action potentials can use different things. Some use calcium, some use sodium. In the case of the action potential in the axon, usually it's sodium. In the case of here, to release the vesicle, the secretory vesicle that has the acetylcholine, this reacts to calcium, right? And that's because this area can't have action potentials. This area can't create action potentials, this like final plate area, right? So it has to use calcium. However, this postsynaptic area also uses sodium to bring the action potential down further into the muscle cell, into the sarcolemma, through the sarcolemma and further into the muscle fiber. Okay, moving on. So steps initiation, once the action potential reaches the sarcolemma, what happens next, right? So again, acetylcholine, which is this, which is the green molecule, it binds to the receptor on the sarcolemma. The receptor opens a sodium ion channel that allows sodium into the muscle cell, into the muscle fiber as well, into the muscle fiber it's also called. Then an action potential is generated because of the influx of sodium causes an action potential. An action potential is generated that travels down the T tubule, which is just a tubule that runs through, throughout the muscle fiber and carries the action potential. Somewhat similar to how axons carry it in our body. The T tubule carries it through this, the muscle fiber, right? Eventually, the action potential moves past the sarcoplasmic reticulum, which is the ER in a muscle cell. And when it moves past it, actually, it depolarizes it as well, the sarcoplasmic reticulum, and the voltage difference, the depolarization triggers a response in the SR, causing it to release calcium into the muscle cell. So normally the SR contains a lot, it's like a storage space for calcium in the muscle fiber. When the action potential passes by it, the voltage difference causes it to release some of the calcium outside, right? It opens a gate, and then calcium can go out of it into the membrane where there's not a lot of calcium, right? It goes down its gradient. The calcium then binds to troponin, which is an important protein we'll cover in the next slide, and it begins the myosin-actin bridge cycle, the actual contraction of the muscle. Okay, so it's actually the calcium that begins it. Okay, and if you want to take a quick picture of this, go ahead. Okay, all right, moving on. Troponin and tropomyosin, right? These are very, very important. So first we cover tropomyosin. Tropomyosin is a filament that is normally bound to myosin binding sites on actin. Actually, if you remember, when I talked about actin here, if you want to visualize it just to help you, in this cycle, I remember mentioned that these blue balls were the actin, right? Which that myosin binds to. This orange line that's running through them is the tropomyosin, right? It's like lines essentially that go through, right? It's a filament that goes through. It's normally bound to the myosin binding sites on actin. So the sites on actin where myosin should attach to, right? And it acts as a blocker. because it covers those sites. It prevents the myosin from binding actin and the formation of the cross bridge. If myosin can't bind and form the cross bridge, there can be no contraction, right? In order to begin the contraction, you need to remove tropomyosin from those sites. Tropomyosin is a protein which has, the way you do that is through troponin. Troponin is a protein which has two different binding sites. It has a calcium binding site and a tropomyosin binding site, right? When calcium is released from the SR, it binds to troponin, right? Which then activates troponin and allows troponin to bind tropomyosin. Troponin can only bind tropomyosin when it's bound to calcium as well. You only have calcium in the membrane of the cell when the axon or the brain or the nervous system tells the cell or the muscle fiber, I want to contract, right? I want you to contract, right? And that activates the troponin and allows it to bind tropomyosin. When it binds tropomyosin, tropomyosin moves out of the way of the myosin binding sites and allows myosin to bind, right? Thus beginning a contraction cycle. If you want to look at this picture, again, these little gray balls are the actin. And if you notice, there's these little blue dots, and these blue dots here as well. Those blue dots are the myosin binding sites on the actin, right? Those are the spots where the actin or the myosin actually attaches to the actin. If you look at here before troponin binding, this little vine thing, tropomyosin, is actually perfectly covering all of these dots, right? It's perfectly covering these dots so tropomyosin so myosin can't bind. However, when this little red thing, troponin binds calcium, it slightly moves out of phase. So if it was like this, it slightly moves out of phase, and now these are all open. It's still there. The tropomyosin is still there next to it. It's just not covering anymore. It just moved slightly, right? And now tropomyosin can bind, and we can have the bridge going. And then when you want to terminate contraction, you just simply do the opposite. Contraction is terminated once calcium is removed from, I shouldn't have said from the SR. It's removed from the membrane into the SR. It actually has to go back to the SR. This sarcoplasmic reticulum has to be removed from the, not the membrane, it has to be removed from the cytoplasm, right? Sorry, somebody asked if I could clarify. The troponin can only bind to tropomyosin when it's bound to calcium. Troponin can only bind to tropomyosin when troponin is bound to calcium. So like I mentioned, troponin is this little like red ball right here, right? And that it's bound to calcium. When it's bound to calcium, it can actually bind to this vine, the tropomyosin, right? When it's not bound to calcium, it can't bind this vine anymore and it can't move it out of the way, right? But the troponin binds to calcium on one side, and then on the other side, it binds to tropomyosin. Okay. All right. Once there's no more available calcium because it's all been shoveled back into the SR, for troponin to bind to, troponin is not bound to calcium anymore because it has to unbind tropomyosin as well. It can't stay attached to it if it doesn't have calcium. Once tropomyosin is unbound by troponin, it returns to its original position, twists back, and blocks all of the myosin binding sites on the actin again. Because even during contraction, the calcium concentration in the sarcolemma and in the cytoplasm is low relative to the concentration in the SR. In order to remove the calcium from the cell, you need active transport mechanisms which use ATP to pump calcium against the concentration gradient back into the sarcoplasmic reticulum, back into the SR. This also goes in, it's also a big factor in why once you die, you have rigor mortis, you become stiff. Again, if you have no ATP, you can't pump the calcium back into the cell. The calcium has to dissipate on its own somehow, but it won't like do that very quickly on its own because of the concentration gradient. Since there's no active transport, the calcium just stays in the cell, and the myosin binding sites are not blocked by tropomyosin. So myosin can actually just stay attached to the actin filaments, right, for a while until either the calcium dissipates or something else gets broken down, right? And eventually rigor mortis does end. Okay, does that make sense? There's actually two reasons that rigor mortis happens. So I know I went through a lot. Do you guys have any questions about troponin, tropomyosin, actin, calcium, the SR, anything before we move on to this big question? Regulation. So most of the regulation is done through calcium. There's obviously a lot more to it, and it's like way more in depth. That's beyond the MCAT, but most regulation, like direct regulation, is done by calcium. You either release calcium through action potentials into the cytoplasm, or you remove it through active calcium pumps out of the cytoplasm back into the SR or outside of the cell, and that's how you regulate contraction. Troponin is not always attached to tropomyosin. Troponin is usually like floating around, if you want to think of it that way. It's hanging around. It's hanging around once it binds to, once it like finds a random calcium floating as well, it binds to it, and then they float together for a bit, and then they bind to the tropomyosin, and then it doesn't reject it. Normally, if it's just floating around and it tries to bind to the tropomyosin, it'll like reject it and it'll just keep floating around, right? And technically, troponin has 3 binding sites, one for calcium, one for tropomyosin, and then it has another binding site, but that third binding site is the I site. But that's I think beyond the scope of the MCAT. That's if some of you may remember if you took like a biochemistry class maybe, or any other advanced cell biology class, but that's beyond the scope of the MCAT. Okay, any other questions before we move on to the big poll question? Okay. All right. So this is a big one, but I really like this question. If you can get this question, I think you understand most of muscle contraction already just by answering this question, right? So I'm going to launch the polling. I'll give you guys a couple minutes. Please look it over, think each answer choice through, and let me know what you guys think. I'll give you guys another 30 seconds to a minute. Okay. So try and get your answers in if you haven't already. Okay, I'm gonna end the poll. Okay, so the majority of people actually picked A and B, right? However, the correct answer is actually C. And let's go over it, right? Let's go over step by step. Number one, dissociation of the myosin head from the actin filament. This goes back to the cycle. And this is true, dissociation of the myosin head from the actin filament. The question says requires binding or hydrolysis of ATP. So essentially ATP really in any form or any function. Dissociation of the myosin head from the actin filament does require binding, right? And like I mentioned, that was the main reason for rigor mortis, because without ATP, you can't dissociate the head from the actin. And so this is correct, right? Attachment of the myosin head to the actin filament, again, it is required by hydrolysis, except this time, right? Hydrolysis of ATP, I said, once ATP binds, it's uncharged, it detached from the head, it becomes uncharged. Then it has to hydrolyze into ADP and inorganic phosphate to become charged and then bind again. Hydrolysis of ATP is required to make myosin charged and to bind it to the actin filament. Conformational change that moves actin and myosin filaments relative to one another. I think this is the one that really made A and B popular because they had three here as well, right? Right, and this is true as well, right? Binding of troponin to the actin filament, nope, because troponin binds due to calcium, right? And it actually binds to tropomyosin due to actin. Release of calcium from the SR. This was an action potential, right? So you don't need that. Reuptake of calcium into the sarcoplasm. Again, yes, because like I said, calcium concentration in the actual cytoplasm is low. So reuptake it is, you need active transport. Active transport requires ATP. So it's this one, right? Right. Oh, I'm sorry. Actually, about this one, you don't need. I made a mistake. It's because it can be loosely bound without. My mistake. It can actually be loosely bound without myosin head to actin filament, right? And for the conformational change, like I mentioned, I believe I went through all of these, right? Yeah. Any other questions? The reason this one's wrong is because you can have a weak attachment to between the myosin and actin filament without hydrolysis or binding of ATP. But the hydrolysis of ATP is required for the power stroke, right? It charges it, which allows it to bind and then perform the power stroke when it comes apart. So that's why 2 is not the right answer. Sorry, 4 is not correct. 4 is not correct because binding of troponin to actin filament. Firstly, troponin binds to the myosin, tropomyosin. Troponin binds to the tropomyosin, and it doesn't require ATP. It requires calcium. Once troponin is bound to calcium, it binds to tropomyosin. It doesn't require ATP. ATP is now required for attachment because it can attach weakly without ATP. right? Without ATP, it can still weakly bind. The reason you need ATP is to actually bind strongly and then perform the power stroke, which you can't do without ATP, right? A weak binding is not enough to perform a power stroke, and you need a strong binding. Yeah. Does that make sense? So attachment, if it said strong attachment, too, but just mention only weak attachment. So it doesn't mention strong or weak attachment. Any other questions? Yeah, so it can attach itself initially weakly. So in the diagram I showed it. So in the diagram, in like the cycle I showed with the picture, it did require the binding to attach strongly to become charged. However, it can technically attach weakly. It'll come off very easily, and then it needs actually the ATP to become charged, actually continue on to do the power stroke. It's kind of similar to how, like in a chemical reaction, in an equilibrium or in any chemical reaction, really, you technically do go forwards and backwards, but overall you go forwards more than you go backwards. So even though it's only showing you go forwards, but really you're going this both ways, right? It's just because it doesn't change, but yeah. I'm not sure if that made it clear, or maybe might have confused a little more. If it confused you, don't worry about it. Didn't forget I said anything, but if you think of it that way, it can attach if it's weakly, but it can't proceed further if it's weakly attached unless ATP is also attached. Unless ATP also is hydrolyzed, I mean. Okay. 2, 4, and 5. Okay. Like I mentioned, 2, attachment of myosin head to actin filament. This does not require ATP necessarily, or hydrolysis of ATP. For example, you can actually be attached to the myosin head and actin filament without ATP being involved. You can also, even when you have ATP, be very weakly bound to it. You just need hydrolysis for attachment, to continue into power stroke. 4, binding of troponin to actin filament. The troponin binds to actin through tropomyosin. However, that doesn't require ATP. That requires calcium to be present, right? You need calcium, not ATP. And 5, release of calcium from the SR. Again, that doesn't need ATP. That does occur for initiation, but it doesn't need ATP. It needs an action potential, which is voltage, or like an electrical source. Oh, this was taken from a real practice MCAT, so I would not be so sure about that, Opal. This is exactly a question that the MCAT would give you, and that's why I like it so much, because this is exactly what they would give you.
Okay, Mark, quick question. So if I was the frequency of number 3, you mentioned it, you said why you skipped it. I mean, I assume it's right because it's in all the answer choices. Is that?
Oh, right, yeah, 3's. Oh, yeah. Also, I guess you could guess, but I didn't even know that. 3's correct because again, you can't have the power stroke without hydrolysis, because you need detachment of inorganic. Remember how you need detachment of inorganic phosphate in order to continue with the power stroke, but the only way to get inorganic phosphate is to hydrolyze ATP, right? Does that make sense?
Yeah, I'm not concerned about,
Yes.
why. It's just that I wouldn't have even bothered reading 3 simply because it appears in all the answer choices. So it's obviously a correct answer. So I would have just focused on the other 1 to 4 remaining. You know what I'm saying? 3's in all the choices. Why would I even bother reading it? Never mind.
Yeah, but yeah, so I actually didn't even notice that until you mentioned it. But yeah. And somebody asked, you said part 5, you need voltage, electricity. Yeah, an action potential is essentially a depolarization, which is you can think of it as like an electrical input, like from your brain, right? It's an electrical input, but it's actually just an action potential that goes through the T-tubule, and when the action potential passes by the sarcoplasmic reticulum where the calcium is being stored, the voltage difference that the sarcoplasmic reticulum experiences causes it to open one of its channels that allows calcium out. Okay. By the way, this is I believe, the last slide. Yeah, this is the last slide. So we are done after this. If you guys have any questions, please feel free to ask away about anything we covered today or anything at all. I'll be on for another, say, 10, 15 minutes to answer questions if you guys have any. Otherwise, we can end off early. And reminder, tomorrow is office hours at 8:30 p.m. Eastern Standard Time if you guys are interested. Okay, if not, I think we can end off.